and strep throat have in common? You might wonder how food and disease can be alike, but the examples given all result from microscopic organisms called bacteria.
So as they say, the question here we have, why do yogurt, cheese, and strep throat they have in common? It's all about bacteria. Bacteria, first of all, some students they might ask, Master, where are they? Where do they live? Bacteria, it's everywhere. On your hands, on your desk, on your earphones, on your mobiles. They live everywhere, even in the sand, air, and water. They don't mind any environment. They can survive in every single environment. First of all, just general information about bacteria. First, bacteria can live in very vast temperatures. That means they don't have a problem if the temperature is hot or if temperature is cold. That's why you can find them everywhere. Second, can reproduce in as little as 20 minutes. That means every 20 minutes bacteria they can reproduce. When we say they can reproduce, bacteria, as we took in the previous lesson, they are prokaryotes. That means they can reproduce asexually. That's why it is very fast. Here just it says one bacteria cell can become 16 million in 8 hours. One bacteria cell, after 8 hours, it becomes 16 million bacteria cells. Don't forget, when we say the word bacteria, we are talking about bacteria cell. First, scientists, they considered one of the ancient organisms. They were found 3 billion years ago. Can survive in extreme conditions, such as without oxygen or without light. Good thing you should know about the bacteria that 99% of all bacteria are helpful. That means you shouldn't worry more on bacteria than on viruses, because we don't have helpful viruses. All viruses are harmful, but some bacteria are harmful. Most, which here says general, just the fact that 99% of all bacteria are helpful. On our body, as you can see on our body, we have good and bad bacteria in humans' body to maintain a healthy balance. You can just here also, let's go quick on the where we can find bacteria. First of all, located on all our body, externally and internally. That means they can survive in our skin. Look at your hands. Here it says number of germs on your fingerprints doubles after using a restroom. So once you go to the restroom, doubles number of the bacteria on your skin. And only 70% of people wash their hands after using a public toilet. Nowadays, of course, you have to keep washing your hands, especially not nowadays. It should be part of your lifestyle. Before having a food, after having a food, after coming back to home from public or outside places, you have to keep wash your hands, despite if you have a coronavirus or there is no virus. This should be part of our lifestyle. Not because nowadays we have coronavirus, you have to keep wash your hands. So it should be done regularly, or it should be one of your lifestyle. So those are the places, or most places, that you can find bacteria. Look at here on the earphone. Wearing headphones for an hour can increase the bacteria in your ear by 700 times. So those are the just general hints about where do you find, or general facts, where do you find bacteria in our body. Is there any question? Guys, do you have any question? Is everything clear? So we can continue to the. Yeah, if you have any question, please just put me on the chat, please. Okay. Structure of a bacteria. Structure of bacteria. By the way, we took this in the previous lesson.
Prokaryote structure and characteristics. Prokaryotes are microscopic, unicellular organisms. They have some characteristics of all cells, such as DNA and ribosomes, but they lack a nuclear membrane and other membrane-bound organelles, such as mitochondria and chloroplasts. Although a prokaryotic cell is very small and doesn't have membrane-bound organelles, it has all it needs to carry out life functions. As with other types of organisms, prokaryotes now can be identified using molecular techniques. By comparing DNA, evolutionary relationships can be determined. Historically, scientists identified prokaryotes using criteria such as shape, cell wall, and movement. Bacterial cells have enough structures, such as DNA, capsule, and flagella to carry out life functions.
So, as you heard in this video, it says that because bacteria, they are very large groups, so scientists, they start to classify them or to put them in a group according to their shape, according to their cell wall, or according to their movement. Why? Because we have, as we said, we have a large number of bacteria. They come in a variety of shapes, variety of cell walls, and each one has different way of movement. As we know, the structure of a prokaryote, especially bacteria, let's go from outside back to inside. First of all, outer membrane capsule. We are going to see later what is a capsule. Under the capsule, they have of course, the green one here. It is a cell wall. This one, cell wall of a bacteria. The blue one, as we said, it is cell membrane, which is one of the common for every single cell. Inside the cell, because prokaryotes, they lack nucleus and they lack membrane-bound organelles, they simply have ribosomes. Simply, they have ribosomes. They have chromosome or DNA. Of course, the empty space, which is a cytoplasm. As you saw externally, bacteria have flagella. Flagella enable them to help bacteria to move. You are responsible for this figure, even though we took it in the previous lesson about the eukaryotes, the comparison of eukaryotes and prokaryotes, but you are responsible for the structure of a bacteria. Don't worry, the PowerPoints will be uploaded on the school website, so you can have it, or you can all have it. Or if you have your books, of course, you can find it, chapter module 17, lesson 1. If you don't, of course, even if you have it or you don't have it, we are going to upload the slides on the school website. So prokaryotic shapes, it might they have a tree. It can be cocci, bacillus, and spirocytes. We are going later back to our PowerPoint presentation, so you can see the shapes of the bacteria.
Prokaryotic size. As prokaryotes lack a nucleus, so their chromosomes are in the cytoplasm. Their genes, in their cytoplasm, are found on a large, circular chromosome in an area of the cell called the nucleoid. Many prokaryotes also have at least one smaller piece of DNA, called a plasmid, which also has a circular arrangement. The chromosomes of prokaryotes are in the cytoplasm. Prokaryotic chromosomes are circular in nature.
So here, just talking about the chromosomes, they call the nucleoids. What is a nucleoid? Here we can, as you can see, area in prokaryotic cells that contain large circular chromosomes. One of the comparison was animal chromosomes, a long linear chromosome which is made up of double strand of DNA. The bacteria chromosome are circular chromosome, of course, again made up of double strand of DNA. Yeah, bacteria. Bacteria, diversity of prokaryotes. Bacteria are microscopic organisms that are prokaryotes. Prokaryotes are divided into two domains, bacteria and archaea. Boys, we are not going to take too much about the archaea, but just for you, just know that archaea here, archaea and bacteria both considered as domain of archaea and the domain of the bacteria both considered to be as a prokaryotes. We are not going to take too much about archaeas. Yeah, bacteria, they have very strong cell wall. Their cell wall is made of a peptidoglycan. We are going to see what are the peptidoglycans. Some have second cell wall. Archaea, just for you general information, thermoacidophiles live in a hot acidic environment where bacteria they cannot live. Halophiles live in very salty environment. That means very high salt concentration, of course, where you cannot find any organism except archaea. Methanogens cannot live in the presence of oxygen. That means a place that there is no oxygen, you can find only archaea and some bacteria, because later we are going to see some bacteria are anaerobic. That means even they don't need oxygen. Archaea, instead of using oxygen, they use carbon dioxide and they produce methane gas. Instead of using CO2, instead of using oxygen, they use carbon dioxide. Instead of producing CO2, they produce a methane gas. Even both are prokaryotes. Some scientists, they find differences between bacteria and archaea. As you know, both are eukaryotes, prokaryotes, but still we can find differences between them. Here it says, cell wall of bacteria contains peptidoglycan, but cell wall of the archaea do not. Boys, don't worry about the peptidoglycans. We are going to see later comparison of peptidoglycans. But keep in your mind, the bacteria cell wall made of a peptidoglycan, but archaea, they don't have it. Second difference, the two groups of organisms have different lipids in their plasma membrane. As we took previously, all membranes are made of a phospholipid bilayer, but even though the phospholipid bilayer in the bacteria is different than composition of cell membrane in the archaea. This is the second difference between bacteria and archaea. They have different composition of their plasma membrane. Third, different proteins, different RNA. According to these scientists, they classified bacteria and archaea to have things, even though they are even though both are prokaryotes, but they have several different things between them. of yesterday, we saw 4 membranes. Okay, today's lesson is about how bacteria they reproduce. Of course, bacteria is a single cell, as you can see in the figure. As you can see here, they are single cell organism. That means they can reproduce asexually. When we say asexual, that means they don't need another partner. They don't need another cell in order to reproduce. It can reproduce by itself. The name of the production in bacteria, we call it binary fission. Let's start and let's see how binary fission takes place.
Reproduction of prokaryotes. Most prokaryotes reproduce by an asexual process called binary fission. Binary fission is the division of a cell into 2 genetically identical cells. Select next to view the process of binary fission.
So
Under ideal environmental conditions,
it says they reproduce as binary fission. What is binary fission? That's definition you can see here. Asexual form of reproduction. Asexual, keep in your mind, that means they don't need another one cell. They don't need 2 cells, only 1 cell. Some prokaryotes, of course, we are talking about prokaryotes, mainly bacteria, in which cell divides 2 genetically identical cells. That means when we say genetically identical, that means copy-paste to each other, exactly as they are, as if you are printing or you are copying something. One bacteria becomes 2 genetically identical bacteria cells without any difference between them. This is, of course, sexual has advantage and asexual has advantage and disadvantage. One of the advantage, if the parent cell is good, that means it's healthy, all products will be healthy. That means all organisms that produce from this mother cell will be healthy. And the opposite is true. If the parent cell is affected, that means if this cell over here, if it has any disease or anything happens, all the coming will copy the same disease. This is the disadvantage of asexual. Why? Because it's copying process. How it works? This is a simple cell. This is simple bacteria cell. Of course, cell wall, plasma membrane, and chromosome. Next step. What happened? What do you realize? What happened to the cell? Or which process has been done? Which process has been done over here? Atom, it's not a mitosis. Mitosis takes place in eukaryotic cell, but mitosis takes place in our cell. We agreed it is a binary fission. Here, what happens? What's the difference between these 2 figures? This is the original cell. This is just beginning of binary fission. Over here, as Rawat mentioning that the chromosomes split into 2. No, the chromosomes copied, not split. When you say split, that means you are dividing. No, chromosomes has been copied. Why? When we say chromosome, we are talking about the DNA. So the first step, they copy their DNA in order to have 2 identical DNA. Third, as you can see here, the cell starts to constrict, or the cell starts to separate. And number 4, simpler, they split into 2 identical cells. That means those 2 cells are genetically identical. They copy paste from each other. This is the binary fission. Okay, to understand much more, Let's go and see video about binary fission. And please tell me if the voice is clear or not on the video.
Bacterial cells generally increase in numbers by the process of binary fission.
Audio is clear. Can you hear the audio?
The cell increases in length while doubling all of its parts, then forms a cross septum and divides to become 2 cells. 2 cells become 4. Those 4 become 8, and so on. This pattern of increase results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in numbers every 30 minutes. Other bacteria have generation times of several hours or even days. During growth of the cell, streptococci and some other gram-positive cocci synthesize new cell walls in the septal region. Rod-shaped bacteria such as Escherichia coli, Salmonella, and Bacillus species synthesize new cell walls over the entire surface of the cell.
So, I think the video was clear. They were like, each species of bacteria, they have certain time. So that's why scientists, they took the average of bacteria cell, which is about 20 minutes. That means every 20 minutes, each bacteria becomes 2. 2 becomes 4. 4 becomes 8. You have to double always, not to add by 2 or not, yes. 2, 4, 8, 16, 32, 64, 128, and so on, so on, so. Double, always double, not time, not plus 2. Yesterday we saw within an 8 hours they become in a million. Millions bacteria cell, and all these cells are genetically identical to each other. That means they have the same function, they have the same shape, they have the same genetic information. To be much easy for you, here we can just simply demonstrate how the bacteria they reproduce. First, simple bacteria cell. First step. Bacteria cell, as you should know that the bacteria, what do they have is genetic. Inside they have circular DNA. Simple, they have circular DNA. The first step, of course, the cell has to grow to increase in size. Not directly they start binary fission. No, the first thing, the cell has to grow or to increase in size. Once they are increasing in size, or once they are big enough, mature enough, the second process is that they copy their DNA. That means here they will have another copy of their DNA. So they have 2 copies of DNA. Of course, boys, when we say copying of DNA, that means exactly the same without any difference between them. Now, second step, so the first cell grows and copies their DNA. The second step, what happens is that, okay, let's make it with the blue so you cannot confuse between them. Second step is what happens to those 2 DNA? They will start to separate. Separate, that means they move to the opposite sides. So first they copy their DNA. Second, separation of their DNA. Step number Step number 3. Step number 3. Those steps are important. Step number 3. Cell membrane will start to constrict. Cell membrane, sorry for the drawing, is not that much clear. So the cell membrane will start to constrict, not separate. Still, it is a beginning of separation of the membrane of the cell, of course. And the step number 4, the last step, simple. The cell will be split into 2 genetically identical cells. Each one, of course, will have a copy of the DNA. This becomes 2. This one again becomes 2. So each cell generate 2 identical cells, and so and so and so. So scientists, simple if you can do it at home. If every 20 minutes they are doubled, so within a day, of course we are talking about 1 bacteria. Of course you will not be infected with single bacteria, you will be infected with many bacteria. Let's say if you are infected with 100, so within 20 minutes 100 becomes 200, 200 becomes 400, and so on. So after an hour, the number of the bacteria will be almost billion. So the steps of binary fission, simple. Cell grows and copies their DNA. Step number 2, DNA separates to opposite side of the cell. Step number 3, cell membrane starts to pinch inward or to constrict inward. This is the beginning of the separation. Step number 4, simple. The cell, bacteria cell, mother cell, this cell splits into 2 identical cells. Those are simple of steps of genetic, the binary fission.
Binary fission can occur quickly, as often as every 20 minutes. If conditions are just right, 1 bacterium could become 1 billion bacteria through binary fission in just 10 hours.
So here, under ideal environmental condition, that means if bacteria, they have specific temperature, or temperature is okay, nutrients are available. So ideal, that means everything is normal, or normal environment. That means without any restrictions of anything. 1 bacteria, so here they gave you again the average time of the bacteria. It says every 20 minutes. Of course they are talking about the average. If conditions are just right, 1 bacterium, this is a singular of a bacteria, 1 bacterium could become 1 billion to 10 hours, just in 10 hours. Okay. Any question on Z conjugation? Yes, this is important. Some bacteria, they undergo another type of reproduction, which is conjugation. And by doing this conjugation, they exchange genetic information. That means they become more diverse. Here it says they become more diverse. Diverse, that means different types, different genetic combination, different. This is the advantage, or this is helpful for bacteria. Of course, this is a disadvantage for us. Okay, why? Because later we are going to see because of conjugation, some bacteria becomes resistant to antibiotics. Okay, let's see this video about conjugation or how the process of conjugation takes place. You can see I think it's clear, right? If there is an audio problem, please inform me. If there is an audio problem, inform me. Sorry. Yeah, let's start. Conjugation process.
Bacterial conjugation is a process of genetic transfer between bacterial cells that requires direct contact between the cells. Many, but not all, species of bacteria can conjugate. Conjugation can occur between cells of the same species or even between cells of two different species. Species of bacteria can conjugate.
As you can see, it says can occur between cells of the same species or even between cells of two different species. So this is an advantage for them. It can be of the same species, for example, rod with rod shape, or cocci with rod or cocci with spirillum. So that means conjugation can be done between all bacteria cells, not only same shape, same type, different species.
Conjugation can occur between cells of the same species or even between cells of two different species. A small DNA circle or plasmid called the F factor is required for conjugation. The F factor stands for fertility.
So as you can see in the figure in front of you, the first bacteria, the first one over here they have a gene or plasmid or a segment of a DNA which is missing in the second one. This one only found in the first bacteria, and here it's missing in the second one. Right here, this one they don't have this gene. So now what's going to take place? Now they will do conjugation. A segment of the red gene or the segment of this DNA will be transferred to the second bacteria. Okay, this is conjugation. They are sharing or they are exchanging genetic material. They are simple exchanging genetic material.
Factor. Strains of bacteria containing the F factor are called F+. Those without it are called F-. An F+ cell, or donor, produces a structure called a pilus to connect with another.
Yep. The one that they have this gene, simple, we call them donor. Donor, the one that will donate, or donor is the one that will transfer this gene to the other one. So donor, the one that will donate or will transfer the gene to the, of course, in order conjugation to take place, we talked about last time about the pili or simple singular pilus, right? Pili, hair-like projections on the bacteria. Main function of it to adhere, to attach to the other surfaces. This was the function of the pili. So in order for conjugation to take place, now they need a bridge. Bridge, that means they have to be connected to each other, simple using their pili or pilus, singular and plural. So pilus will connect two bacteria together. Once they are connected, now the conjugation process will take place.
Recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a protein assembly called the relaxosome, which associates with the strand to be transferred, or the tDNA strand. Accessory proteins of the relaxosome are released, but a portion of the relaxosome called the relaxase remains attached to the tDNA. This tDNA relaxase complex is recognized by a coupling factor and transferred to the exporter, a complex in the F+ cell that is contiguous with the pilus. The exporter pumps the tDNA relaxase complex into the recipient cell. Once the entire tDNA molecule is transferred to the recipient cell, relaxase joins the ends to make a circular DNA molecule. As the tDNA is transferred to the recipient cell, it is replicated to become double-stranded. In the donor cell, the F-factor DNA was also replicated to become double-stranded. This actually occurred as the tDNA was being transferred to the recipient cell. In the end, both cells wind up with a complete double-stranded copy of the F-factor. Their connection through the pilus is released, and each is now an F+ cell that can go on to conjugate with other cells.
So, simple process. So simple, what you are responsible for it is just bacteria. In order to do conjugation, they will use a pilus. Over here, as you can see, the green one. They will use a pilus just to connect two bacteria together. Once they are connected, segment of DNA, as you can see in front of you, segment of DNA, as you can see in front of you, the red one, simple will transfer all the way to the second bacteria or to the next bacteria through the pilus. As of course, you know that the bacteria is double strand. So this one, as you can see here in front of you, is double strand. So one of the strand will be simple transfer to here. Later on, it will copy itself. It will be double-stranded DNA. So of course, when we say DNA, don't forget that this has some genes that are responsible for different functions. Maybe to become resistant or to form or for many other chemical reactions, okay? As you can see here, the transferring of DNA from one cell to the other through the pilus. Here they have single strand of DNA. Now later on they will copy their DNA in order to be double-stranded DNA. Now the second one, as you know that the bacteria can do binary fission. If this cell will do binary fission, simple will copy and paste itself. That means will copy both genetic material, which are the blue one, will copy both the blue one and the red one. Okay, so this one will do binary fission. Here they will have a red DNA, of course, and the same what the blue one. Why? Because binary fission we said to produce two identical bacteria cell. Now what happens? This one, they have extra chromosome or extra gene that was absent previously. Any question? And of course, it will continue forever having this segment of the gene in them. Yes, Adam. Just unmute yourself. Just unmute yourself.
Yeah, hi, sir.
Yes, go ahead.
So, sir, we don't need to memorize the movement of the relaxase.
No, nothing. Just the idea I told you, simple.
So the idea is that we have two different. So we have the same type of bacteria, or the different, or a
yes.
different.
Or what is conjugation, or what is the process of conjugation? Simple. Bacteria, they use a pili or pilus just to adhere as a bridge to connect to different species of bacteria. Once they are connected, they are exchanging genetic material. This is the whole idea.
Do they exchange a small strand of the genetic material or the
Yes, a
whole cell?
strand. No, the whole thing is common. The whole thing is
Yeah,
common. They have it. Without DNA, there is no cell. You cannot consider a cell.
they just share a small strand.
Yes, they are sharing. They are simple. This one has no this gene, so they might share extra genes. Extra genes increases the diversity of bacteria. They become more diverse. More diverse, that means more different types, different properties.
Oh, okay.
This is all you should know about conjugation.
Thank you so much, sir.
Welcome. So, as we said, scientists last time they classified bacteria according to their shape, according to the cell wall, right? It can be positive or it can be negative. I mean, gram positive, gram negative. The third way that scientists they classify the bacteria is simple: how they obtain food or according to their nutrients. As you can see here, scientists classify the prokaryotes to be heterotrophs and to be auto. Heterotrophs, simple, we took them in nitrogen cycle, we took them in carbon cycle. They are decomposers or they are saprophyte. They are decomposers. They feed on that organism, that animals or that plants. By secreting enzymes, they digest and they absorb their nutrients. Those are heterotrophs. Simple, they feed on that organism. We took them in nitrogen and carbon cycle. What about autotrophs? As you know that plants are autotroph, that means they make their own food. This is what we took in photosynthesis. Plants are auto, they make their own food. So prokaryotes or bacteria can be divided into two groups. It can be photoautotrophs. Simple. Photo means the light. Autotroph, that means they make their food using a light, or we can call it photosynthetic autotroph. Same as a plant. They use the sunlight to make their own food. Or chemoautotrophs. Chemo comes from the word chemicals. That means instead of using light energy, they use the energy that is found in chemicals. Okay, so those are the classification of bacteria according to their nutrients. Heterotroph and autotroph. Autotroph divided into two other groups, photosynthetic autotroph and chemoautotrophs. One more thing you should know, we have aerobic and anaerobic bacteria. So here, anaerobics or anaerobes are bacteria that they don't use oxygen. You can find them in your slides. Anaerobes or anaerobic bacteria, they are prokaryotes that they don't use oxygen. And we have aerobic or aerobes, they require oxygen to grow. There is one intermediate, we call them facultative anaerobes. That means they grow either with or without oxygen. So we have bacteria that they need oxygen, simple like animals, animal cell. We have another group of bacteria that they don't need oxygen to grow. We have the third group, they don't mind if there is or there is no oxygen. They can survive with or without oxygen. So those are the classification of bacteria according to their nutrients or according their needs. So here, as you can see in front of you, as you can see in your slides, photoautotrophs, simple, they carry on photosynthesis. One of the simple or one of the famous bacteria that they do or photosynthetic is cyanobacteria, and some scientists they consider or cyanobacteria were responsible for production of our oxygen that we are having or to form our ozone layer. Chemoautotrophs, we said they use chemicals. Simple, they break down and release inorganic compounds such as nitrogen or sulfur. like nitrites, nitrates, ammonium. So they feed on those nitrogen cycle, like sulfur, H2S, yes. Chemoautotrophs, yes, those are organic compounds, so they feed on organic compounds, inorganic compounds. And here, as you can see, we have aerobes and anaerobes. Aerobes, aerobic bacteria, they need oxygen. Anaerobes, anaerobic bacteria, that they don't need oxygen. Boys, you are only responsible for cyanobacteria because this is famous, cyanobacteria. Only you are responsible for this example, cyanobacteria. Even in your book, they didn't mention other groups of bacteria. So anaerobic bacteria do not use oxygen to grow. Simple, they can survive without oxygen in anaerobic environment or environments that there is no oxygen. So those are the classification of bacteria. Habib, we mentioned last week that PowerPoints are on the website. Okay, the PowerPoints are on the website, the school website. It's simple, you can go and download from MIS portal, and by the end of this week I'm going to upload you the slides of virus. And the end of this week, once we are done with the bacteria, I will upload you the slide of virus. So those about classification of bacteria according to their nutrients, or according to the food that they need to survive. By the way, this is your ebook that you can see in front of you. This is exactly the same as your book. So heterotrophs, photoautotrophs, and chemoautotrophs. Photo, simple light, using a light. Chemoautotrophs, they feed on chemicals, inorganic compounds such as ammonia, nitrogen compounds such as ammonia, and hydrogen sulfide, H2S. Okay, so chemoautotrophs and photoautotrophs. Autotrophs, that means they make their own food. They are not heterotrophs, they are autotrophs. Simple, they make their own food. Questions? Yes, gram positive, gram negative, you have them in your book, and it is included in the, or I will modify and I will send it to you again. Survival of bacteria. Survival of bacteria. Sometimes you know that we have harsh environments or sometimes we use heat to kill bacteria. Simple, by heating or by preserving food in a cold environment in order the bacteria not to survive. Again, some bacteria, they can survive in those harsh environments. Okay? Some, of course, but not all of them. How? Simple, by forming endospores. Simple, by forming endospores. What are endospores, or how this process takes place? We are going to see how bacteria are responsible for forming endospores. Any question on so far? Any questions so far? Okay. Let's continue the process of endospores. Let's continue the process of endospores.
Reproduction of prokaryotes. Most prokaryotes reproduce by a
Yeah, endospores.
survival of bacteria. How can bacteria survive if their environment becomes unfavorable? They have several mechanisms that help them survive environmental challenges such as a lack of water, an extreme temperature change, or a lack of nutrients.
So some bacteria, they can survive environment that there is no water or without water, drought, extreme temperature, that means high temperature, or lack of nutrients. So in order to do that, some they have a mechanism or they can form endospores. So just see what are the endospores definition. As you can see in front of you, endospores, dormant bacteria. That means bacteria is alive, but it is not active. It's not reproducing, it's not growing. Simple, alive. Dormant bacteria cell able to survive long periods of time without being damaged. We call it a dormant. Boy, same as a tree during the winter. Plants or some trees during winter they become dormant. Dormant, that means they are alive but they are not growing. Dormant, they are alive but they are not growing, they are not reproducing, but they are still alive. They are not dead.
Endospores. When environmental conditions are harsh, some types of bacteria produce a structure called an endospore. The bacteria that cause anthrax, botulism, and tetanus are examples of endospore producers. Endospores are resistant to harsh environments and might be able to survive extreme heat, extreme cold, dehydration, and large amounts of ultraviolet radiation. Any of these conditions would kill a typical bacterial cell. Because a bacterial cell usually produces only one endospore, this is considered a survival mechanism rather than a type of reproduction.
So simple, this is called survival mechanism rather than reproduction. And each bacteria or some bacteria, they can form only one endospores. And these endospores will let them to survive in this harsh environment. Questions so far?
Select next to see.
Any question so far? Now we are going to see the mechanism of how bacteria forms endospores when the temperature, when the conditions becomes unfavorable. But of course, keep in your mind, not all they do that. Some bacteria, they do that. Some bacteria, they do that. Not all bacteria, they can survive in harsh environment. Mainly simple, we can heat the bacteria or we can heat the food in order to get rid of bacteria. Endospores, Sultan, or the others, yes, endosperm, it is a survival mechanism. Now you are going to see bacteria will take care of its genetic material, will cover and protect its genetic material when conditions becomes unfavorable. Why? Because genetic material, they need genetic material in order later, when the conditions are favorable, in order for them to do binary fission or to start to reproduce. And of course, when they form endospores, endospores can survive or can stay alive for years, for decades. Not only 1 hour, 2 hours, or once while you are heating. No, endosperm can survive for many years. Okay, let's continue and see the mechanism. Maybe you will understand it much better. So first, as you can see in front of you, this is simple bacterial cell.
How a bacterial cell produces an endospore.
So bacterial cell grows under favorable conditions is called vegetative cell. Normal, when conditions are favorable, they can survive normal. Okay, question, what happens when condition becomes unfavorable? That means when we have high temperature or when there is no nutrients or when there is UV light, anything that harms the bacteria. So as you can see, the sporulating cells, those are the cells that forming what? Spores here. This is in front of you. The bacteria, when bacterial cell exposed to the harsh environment, they form spores. Still, the spores is inside the cell. This is sporulating cells. They start forming spores when they are exposed to the harsh environment. Okay? What is the function of these spores? Of course, as I said, they will protect or they will copy bacterial cells chromosome and a small part of the cytoplasm. So spores takes the bacteria whole chromosome and part of the cytoplasm. Why? Because it's important of bacteria to survive, it's their DNA. So this is the aim of sporulating cells, to cover and protect their DNA. As you can see inside the spores, they cover their DNA and some cytoplasm. Next, step number 3. Here, as you can see, the bacterial cell itself might die. Why? Because the environment is harsh enough. That means high temperature or UV light or acid or base or pH level. Bacterial cell itself, bacteria is dying. As you can see, bacterial cell here is dead because they cannot tolerate the temperature, or the cell wall of bacteria will be damaged and the cell itself will die. But what happens? Endospore remains. Endospores can be tough as dormant cell and can survive with extreme environmental conditions. So the cell itself dead, but of course the endospore can survive this harsh environment. And of course endospore contains genetic material, or the endosperm covers and protects their DNA. Any questions so far? Any question till now? So when conditions are unfavorable, bacteria, they will form endospores. Endospores will cover and protect the DNA and some cytoplasm. The cell will die. The cell itself will die, but these endospores will survive, or they can survive this harsh environment. Step number 4. When environment conditions becomes favorable, that means back to normal temperature, back to normal nutrients, back to normal conditions, what happens? Endospore grows or germinate into new bacterial cell. As you can see, this is the endospore that survive this or tolerate all this harsh environment. Now, when the temperature backs to normal, when the environment backs to favorable, what happens to the bacteria? Simple, they start germinating. Germinating, that means start to grow their cell wall and cell membrane. Notice that the capsule and the cell wall are start to grow. Here you should notice that the cell wall and cell membrane will start to grow. Of course, who will give this information for the cell wall and cell membrane to grow? From where they will get their information? Yes, from the DNA, from the chromosome, yes, from the DNA or from the chromosome. That's the aim of endospore. Endospore will take care of their DNA because all genetic information is over there. The cell will die, but their genetic information is preserved or covered and protected. When the conditions are favorable, the DNA will give information in order to start forming new cell wall and new cell membrane. And of course, the bacterial cell will grow. As you can see here, the bacterial cell or chromosomes starts producing pieces of new bacterial cell. Inside it, of course, they will have their DNA, everything back to normal. The endospores disintegrate because they don't need any endospores anymore. No need for them because conditions back to normal are able to survive for a long periods of time.